Hi, good afternoon. I'm Ricardo Hayashi, veterinarian, visiting scholar, and PhD student at the University of Illinois at Urbana-Champaign. Today, I would like to talk about the issue of salmonellosis and food safety from chicken products and describe how we can develop new products like probiotics, for example, against this disease for poultry and for public health. Let me ask you something. Do you like chicken meat? I love it. And I assume there's a big chance you like it too. Chicken meat is considered the second most consumed meat all over the world, just after pork meat, which is the first one. According to the FAO, Food and Agriculture Organization of the United Nations, we consumed around 45 kilograms per person per year just here in the United States, and around 30 kilograms per person year as average of the world. Chicken meat is one of the most affordable meat for consumers, about $1.45 dollars per pound. It's considered healthy once that chicken meat contains about 2 to 3 times less polyunsaturated fat than most types of red meat. And of course, it is an amazing protein source. Well, and how can we explain the evolution of the chicken through the years? You can easily notice the difference between the broiler chicken from 1947 and our modern broiler chicken. Observe that the shape of the body. The chicken from 50s could achieve about 2 pounds in only in 20 weeks, which is a long period. In contrast, our modern chicken broiler can achieve more than 9 pounds in only 6 weeks. It's amazing, right? Now you might be wondering how we can accelerate the chicken evolution. Hormones? Magic? No. Essentially, this is a result from years and years of qualified research on those 4 different areas. Genetics, to select those genes in animals which has the best characteristics. Technology, especially for environmental conditions. Nutrition, selecting the quality of feed ingredients and the perfect diet balance for the animals. And a lot of research in biosafety and control of diseases. So veterinarians, animal scientists, biologists, geneticists, engineers, a lot of people involved in this area have been worked hardly to guarantee an animal with the highest performance, welfare, meat and eggs quality for the consumers. Okay. But everything is not so pretty as it looks like. This artificial evolution can be dangerous sometimes. As I said before, when we select genes in animals with the best profile and characteristics, we improve some desired characteristics. For example, we choose to gain muscles in short times, low fat and high egg production, for example. However, at the same time, some undesired characteristics might happen. Now, the modern broiler has a weak immune system. In other words, what I'm trying to say is that those animals cannot respond properly against diseases. Besides that, we figured it out that the gut microbiota means the total population of bacteria in the intestine, has lower diversity of microorganisms. In other words, when we have a high diversity of microorganisms, for example, we have a high number of good bacteria, which can develop a good protection against bad bacteria and diseases. As a consequence, the modern broiler has a low immune response and low gut microbiota diversity. And unfortunately, we are facing numerous bacterial diseases like these: Salmonella, Campylobacter, E. coli, Clostridium, Staphylococcus aureus, and Klebsiella, which is a risk for the chickens and for the final consumers as well, which are us. That brings us to talk more about Salmonella. I'm going to focus more about Salmonella. Salmonellosis is an infection caused by Salmonella bacteria. Most people infected with Salmonella develop diarrhea, fever, vomiting, and abdominal cramps 12 to 72 hours after the infection. Every single year, Salmonella is estimated to cause 1 million foodborne illness in the United States, with 19,000 hospitalizations and almost like 400 deaths. Infections are usually contracted from sources such as poultry, pork, beef, and fish, seafood. If the meat is not prepared very well or is infected with the bacteria after preparation, after cooking. We cannot forget the eggs and eggs products, which can be an important Salmonella source too. We can think about numerous tools to avoid or decrease Salmonella in chickens, for example, organic acids, vaccines, biosafety, and others. One of these tools is probiotics as a feed additive. Probiotic, according to the FAO and World Health Organization, are living microorganisms when conferred in sufficient amount on the host, will render beneficial effects on the health, even animals or human beings. Probiotics, good bacteria, for example, Lactobacillus, Streptococcus, Enterococcus, Bifidobacterium, and Bacillus, which can protect the host with different ways. These good bacteria can secrete a lot of defensins, bactericins, can increase the immune response, reduce the pH of the gut, compete and fight against pathogenic bacteria, which is considered the bad bacteria. It's a natural and safe solution instead of using antibiotics, for example, as growth promoters. Well, and how we can define those good bacteria and maybe develop a safe probiotic for broiler chickens? We may have found the solution in the chicken's ancestors. Feral chickens are derived from domestic chickens. It's a mix of Gallus gallus domesticus and red jungle fowl, who have returned to the wild. As you can see in this picture, it's an amazing picture. This is a feral chicken in Bermuda. We chose Bermuda because there is a huge population of those feral chickens, and also because they live in an island. So it was a perfect experiment with a homogeneous population. They seem to be stronger, very resistant, and well adapted through all different environments. So now look at these graphics. They represent differences in phylum and family diversity of microbiota between the modern broiler chicken and the feral chickens. It's very clear to notice that feral chickens have a huge diversity of gut microbiome, especially in firmicutes, as you can see here, which is the phylum which includes acid lactic bacteria, which are potential probiotics for the future. Well, after we got the samples from feral chickens in Bermuda, we started this in vitro screening for potential for probiotics at Animal Sciences Laboratory. This is a kind of new methodology to guarantee the development and safety of future probiotics for human and animal health. It starts with the isolation of the colonies from feces samples using a selective media. I used MRS for lactic acid bacteria. So after this part, we identified those isolates. With PCR, used the 16S RNA method. Different bacteria can be selected for the acidic pH tolerance. It's the third part, and biosalts tolerance too. And enzymes resistance like amylase, proteinase K, and trypsin, and other enzymes can be tested, and they will be analyzed by antibiotic susceptibility and using 5 different antibiotics. Aggregation tests is useful to measure the adhesion of the bacteria with the enterocytes. And after this, the selected isolates can be tested for antimicrobial activity, which is an important part of this screening test, against numerous types of Salmonella, for Salmonella enteritidis, Heidelberg, Typhimurium, and other pathogens. And if we have, in the end, if we have some potassium probiotic with successful in vitro results, the next step is going to be the in vivo test, which we're going to evaluate the probiotic on broiler chickens challenge with different types of Salmonella, and we can measure the performance, the intestinal health by histopathology, immune response, and the control of this bacteria in the gut. Well, here, now we're going to show some pictures about results about the pH tolerance, for example. It's possible to notice that Enterococcus durans, for example, and Hirae, they show the resistance even in pH 2 or 3 for 3 hours. The same happened in the biosalts tolerance test. All isolates show some kind of resistance in biosalt at 1% after 24 hours. It's an amazing result, especially for 24 Enterococcus durans and 42 Enterococcus durans, which were capable to grow even in high biosalt concentration, 1% of biosalt concentration. To recap on the main point of the acidic and the biosalts tolerance, we can observe in this table the result for all isolates, for all 8 isolates. The 40, as you can see here, 40 Enterococcus faecium and 42 Enterococcus durans show the best results in these assays with the great resistance profile until now. So moving on to antibiotics susceptibility, we tested all isolates in 5 different antibiotics: ampicillin, streptomycin, chloramphenicol, erythromycin, kanamycin, and tetracycline. All isolates showed low susceptibility for streptomycin and kanamycin, and all good results for ampicillin and tetracycline. So the next steps after we finish this part of the screening test is going to be the enzymes resistance, the aggregation test, which is going to be measured when we're going to be measuring the adhesion of the probiotics with the enterocytes, antimicrobial activity, the challenge against Salmonella in vitro, and the end of the experiment, we're gonna perform the in vivo test. So for conclusions of this presentation, Salmonellosis continue to be a recurrent public health problem over the past decades in all over the world. So it's a huge problem of not only for animals but for human beings too. Probiotics can be an efficient tool for Salmonella control in poultry. Feral chickens can be a source of new microbes and potential probiotics due to the high microbiota diversity. And a new probiotic screening methodology has been developed in our team, in our lab, with promising results. Thank you very much.